Thank you very much. Well, good afternoon, and I would like to thank the organizers for selecting my abstract for an oral presentation. So it's a real pleasure to share with you some of the science we've been doing for years. So I've been studying C. difficile phages for 20 years now. We are also exploring other phages in my lab, but C. difficile is a core research topic in my group. So oops. Okay, so it all started about 20 years ago. I was doing my postdoc in Sylvain Moineau's lab at Laval University, and we were right in the middle of a big outbreak of C. difficile associated diarrhea. And as you can see here on these statistics in the province of Quebec, we were just having a lot of cases. And also in the US and in North America in general, but in Europe, a lot of cases were also occurring because of the emergence of an epidemic strain that was resistant to fluoroquinolones. And so people were getting the infections in hospitals. They were, the mortality rate was high. And this is when physicians started to do fecal transplant to try to save some of these patients. And at that time in Sylvain's lab, we were trying to get rid of phages because they were killing good bacteria. But in this case, I was thinking, well, could phages be used for therapy? So I was not too much aware of phage therapy at that time, but I started looking in the literature and I found that, we didn't know much about C. difficile phages. So I thought it would be a good topic to start my career. So you have to know that C. difficile infection occurs in individuals that have a disrupted microbiota. So for those who are not familiar with C. diff, I will just go through a quick overview of the cycle. So people get infected by spores of C. diff that can be found in the environment, also in hospitals, of course. And these spores need to germinate and colonize the gut to produce toxins and cause disease. But in normal, healthy individuals, the gut microbiota prevents the germination of these spores through the metabolic modification of bile acids. So bile acids are used as germinants to favor germination of spores. And so in a normal microbiome, these bile acids are just converted into secondary bile acids that prevent germination and spore outgrowth. Whereas in patients that receive antibiotics, their disrupted microbiota is a favorable environment to allow these germinants to act on C. difficile spores. And this is where the infection starts. Now, the first line treatment for C. difficile infection is to give antibiotics. So you can quickly imagine that giving further antibiotics will just further disrupt the microbiota. So these patients, about 25 to 30% of patients will eventually have a relapse of the disease. So that means additional antibiotic treatments and a higher risk of having another relapse. So it turns out to be a cycle of recurrent diseases. And one way of getting out or breaking this cycle is to perform fecal transplant. But we don't know exactly the long-term impact of fecal transplant. So that's why at some point we need to have more specific treatments to target these C. difficile patients in a more precise way so that the microbiome of these patients can recover without the need to use antibiotics. So I was 20 years ago, I was young, hopeful, and very enthusiastic. So I thought, wow, it should be easy to isolate some C. difficile phage and do phage work on C. diff. But I quickly realized that it would be more complicated than a simple walk in the park on a Sunday afternoon because, first of all, I just, despite many attempts, I couldn't find any strictly lytic phage out there. And I'm not the only one. Other labs in the world that tried to isolate strictly lytic phages failed to do so. So all the phages that we know right now are temperate. And for therapy, of course, we pretty much all agree that we should go for strictly lytic phages, although with the phage antibiotic synergy, we think that we could eventually use temperate phages as well if there's a good synergy with antibiotics. But basically, we should target for strictly lytic phages. So one way to get around this problem is to genetically engineer phages to make them strictly lytic. And I will not go into detail of that today. So this is a project that Andrew Umansky, a PhD student in my lab, is working on. And he's currently working on the creation of cocktails of strictly lytic phages and engineered phages. So stay tuned. There's a manuscript in preparation and we should submit soon for publication. So what I want to talk about today is about the host range and how we study the host range C. difficile phages, because we need to understand how these phages interact with the cell surface of C. diff to be able to design the best cocktails for therapy eventually. So today we'll present the work of Alexia Royer, a PhD student in my lab that just finished a couple of weeks ago. Alexia was in co-supervision with Olga Sutovina from the University of Paris-Saclay in Paris. So the work I will present has been just published a couple of weeks ago, so it's all known material if you go in the publication. So for years we were wondering what kind of receptor C. difficile phages were binding to start the infection. And part of the answer came with the work on difficins, which are phage tail-like bacteriocins that I have observed during my postdoc in Sylvain's lab. So these are the kind of particles that I was seeing. So these particles are similar to R-type bacteriocins that were characterized in Pseudomonas aeruginosa. And a company based in California named Avid Biotics were interested in these particles. So we collaborated together. And then a collaboration was established with C. difficile cell surface specialist in UK, Robert Fagan. And it turns out that these phage particles act as a phage tail, like a contractile phage tail. They bind to the cell surface through interactions with the receptor binding protein on the cell surface receptor. And then through contraction, these tail nanomachines will just puncture the cell surface, leading to cell death. So these strictly lytic particles are very interesting because they allowed Rob Fagan's group to isolate two spontaneous resistant mutants. And upon sequencing of the genome, they found that these two mutants had a single point mutation in the gene coding for SLPA, which is a cell surface protein found at the surface of the peptidoglycan layer, and that creates the protein coat. So SLPA is produced as a pre-protein. It's secreted and then cleaved by a protease at the cell wall interface. And then two fragments are generated, one large molecular weight fragment or high molecular weight fragment that is attached to the cell wall, and then a small or low molecular weight fragment that is attached to the high molecular weight fragment. So if we extract cell surface proteins and we run these on SDS page, we can see these two major bands occurring, which represent the high molecular weight and low molecular weight fragments. And as you can see in the two mutants, FM2.5 and FM2.6 mutants, we don't have these major bands. So essentially, the cells are devoid of a cell S layer. So since the bacteriocins are very similar to phage tails, we were wondering if these phages, these mutants could be resistant to our phages. So we had the strain from which the mutant was derived is an epidemic strain called R2291. And we already had 3 cyphophages that were able to infect the wild type strain. So we tested the mutant. And as you can see here in the spot test assays, the mutant strain was completely resistant to these cyphophages. So it suggested that cyphophages, although they are structurally different from the difficins, they were still binding to the S layer as their receptor. Now, there are at least 13 isoforms of SLPA that have been characterized so far, and if you can see by the alignment here, the high molecular weight region is more conserved, whereas the low molecular weight region is a little more diverse. And here is the crystal reconstitution of the surface. So you can see that you have a low molecular weight fragment sitting on top of the high molecular weight fragment. So this creates a very organized and symmetrical structure on the cell surface. So we decided to use that SLPA mutant strain and complement the strain with different copies of the different isoforms of SLPA and see whether this would change the susceptibility to phage infection. So we conjugated a bunch of the 13 different isoforms of SLPA into the mutant and then performed phage infection assays, absorption assays, and EOP assays. So these results were published 2 years ago in Microbiology Spectrum. So we have found that depending on the isoform that we reintroduce in the mutant strain, we could in some cases recover infection by some of our 3 cyphophages. But more interestingly, we were able to now infect that strain with phages that normally don't infect that wild-type strain. And these are myophages. So it's very interesting. And some of these myophages were able to recognize 2 or 3 different isoforms of SLPA. And some SLP isoforms seem to be more frequently or easily detected by several phages from the tested collection. So that further supported the fact that SLPA and the S layer is a very important receptor for many phages. Now, we were wondering, where does the phage attaches? Since the surface is made of valleys and mountains, where did the phage attach to? So our collaborators in England also generated a mutant strain where they have removed part of the low molecular weight fragment that we call the D2 domain, which sits on top here of the surface, and they removed that portion that led to a S layer that is kind of shaved in some sense. So we got the mutant and we tested it against our cyphophages. And as you can see here, 2 of the 3 cyphophages were no longer able to infect that mutant strain, but 1, the phage CD111, was still able to infect. So that suggests that these 2 phages somehow bind or need to bind to the D2 domain, whereas phage CD111 doesn't have to. We also created similar domain 2 deletions in 3 additional isoforms of SLPA. And as you can see here, only a couple of phages were still able to infect the deleted isoforms, and other phages were not able to infect. So that suggests that some phages need the D2 domain and others don't. So that suggests that they might interact in deeper areas of the S layer, and they don't require the top surface to engage or cause infection. Now we were wondering, for those who were still interacting with the D2 deleted version, were they interacting with the D1 domain or the high molecular weight fragment, or both? So to try to address this, we decided to create hybrid version of SLPA by combining the high molecular weight fragment that is, part of an isoform that is detected by a phage, and combining it with a low molecular weight fragment from another isoform that is not detected normally by the phage, or vice versa. So we created a series of hybrid versions with these squares representing the different isoforms, and at the bottom the high molecular weight fragment, and on top the low molecular weight fragment. And so we created these hybrid isoforms and then performed infection assays. And as you can see here in the spot test assays, we found a number of phages that were capable of infecting these hybrid versions. So that gave us more insight into which fragment of the S layer they were interacting with. Also, we tried to, for those phages that were requiring the D2 domain, we tried to see if we could kind of refine our understanding and delete portion of the D2 domain to see if one of these were involved in interaction with phages. To do this, we looked at the predictive secondary structure of the D2 domain, and we made deletions in specific regions in order to avoid disrupting secondary structures. then we expressed these variant forms in our mutant. And unfortunately, when we look at cell surface extractions, we couldn't find any of the low molecular weight fragments occurring in these cells. But interestingly, we still found the high molecular weight fragment. And our collaborators in UK, they have tried several times to express only the high molecular weight fragment of the S layer, but without success. And here we were able to get these high molecular weight variants. So we decided to do a step back and then create a clean high molecular weight version where we deleted the complete low molecular weight fragment, including the D1 and D2 domain. And we kept only the low molecular weight and high molecular weight interaction domain, which is the site of interaction for the protease that processes the protein during the secretion. So we thought that maybe keeping this would be the key to be able to express only the high molecular weight fragment on the cell surface. So we were successful, and we were able to express only the high molecular weight fragment of SLPA type 4 and 8. And unfortunately, when we did the infection assays, none of the phages tested were able to infect these variant forms. So this suggested that they needed something else from the low molecular weight fragment to be able to infect. Now, just to summarize, we did a series of infection assays with these different variant isoforms, with different phages. We tested adsorption, spot test, we did EOP values. So this is a lot of work done by Alexia and Emeline, especially since everything has to be done within inside the anaerobic chamber. So imagine even the soft agar overlays and spot tests and adsorption assays have to be done in the anaerobic chamber, otherwise it's a mess. So that's a lot of work from these two students. So these results, as I said, were published in PLOS Pathogens a couple of weeks ago. So from all these data, we were able to draw major conclusions, and we think that there are three different scenarios that are occurring. The first scenario is where adsorption is done through the high molecular weight fragment, but portion of the low molecular weight fragment, in particular the D1 domain, is required for infection. So this is the case, for example, with phage MMP4 that binds the wild type SLPA type 4, SLPA type 8. It also binds the D2 deleted version. It adsorbs to the high molecular weight fragment only, but it's not able to infect. But if we provide the high molecular weight fragment from SLPA type 8 plus the low molecular weight fragment from SLPA type 4 that it normally doesn't recognize, we do have infection. So that suggests that there is a conserved region in the low molecular weight type 4 that is somehow conserved enough to allow infection. The second scenario is exemplified by phage 146. So this phage normally binds to the wild type 4 SLPA, but it doesn't bind to any of the deleted forms. So it seems to interact with the D2 domain, because if we delete the D2 domain, we lose infection. It also infects SLPA type 11, the wild type form, but it is not able to infect any of the hybrid versions that we tested. So it seems to require both the high molecular weight and low molecular weight fragment from the same isoform. There's an exception, though, with SLPA type 8, because this phage phage 146 is able to infect the hybrid version where we have the high molecular weight fragment from SLPA type 4 and the low molecular weight from type 8. So somehow, even with low identity, there's some conserved structure or amino acid residues that allow infection. And for those who are attentive, you will see that there is no adsorption here. So in fact, we have also observed some adsorption delays in our normal assays. So that makes adsorption tests a little more challenging. And lastly, the third scenario is exemplified by phage MMP02. So this phage binds to SLPA type 6, the wild type form, doesn't bind the D2 deleted form, but it does bind a hybrid version where the low molecular weight 6 is fused to the high molecular weight of type 4, which normally is not recognized by the phage. So that suggests that this phage is binding and interacting with the D2 domain. This is a similar situation with SLPA type 8. And for type 10, the phage binds to the wild type isoform, the D2 deleted form, but when we test the hybrid versions, we don't have any adsorption or infection. So this phage seems to require the high molecular weight and probably the D1 part of the low molecular weight fragment from that SLPA. So to wrap up, our study allowed us to make important conclusions. First, that SLPA is an important receptor for different phages, including cyclophages and myophages. Interestingly, we also found that the SLP isoform that are detected by cyclophages are different from those detected by myophages. So that is also an interesting observation. We also found that SLPA is determinant for the phage host range. So some phages will bind to one or a few SLPA isoforms. And if we look at a broader bacterial population, we have seen that specific isoform, when it's present in a strain, it is well conserved in a group of strains that have the same isoform. We also observed that the D2 domain is important for some phages, but is dispensable for others. And based on all the interactions that we have done, we were able to delineate three main interacting scenarios. So all in all, this work will allow selecting and engineering phage cocktails that will target different SLP isoforms or different regions of SLPA. So that if there's a mutation occurring in the SLPA in the part that is recognized by one phage, another phage could eventually bind to another region of the S layer, therefore allowing infection. But the S layer is just half of the picture because we also need to look at the phage counterpart, so how the phage interacts through its receptor binding proteins. And this is also work that is ongoing in my lab. And finally, understanding how phages interact with their receptor will facilitate the future engineering of phages that will target specific C. difficile isolates. And hopefully we will be able one day to develop phage therapy against C. difficile. So with that, I would like to thank the students that were involved in the project, especially Alexia Royer, who, as I said, just finished her thesis a couple of weeks ago. Also Andrew Umensky, Min Dion, also former lab students that contributed somehow to this project. My collaborator in Paris, Saclay, Olga Stolina, Joanne Pelletier, people from University of Sheffield, funding agency, especially CIHR. I got a CIHR fund this year. I'm pretty happy with that to continue that project. So with that, I will take any questions if you have any. Thanks.